Oh, long day. How you guys feeling about that? You don't care if I had a long day or not, do you? I know you. Actually, you probably do. But in any event, how's your studying coming along? Coming along. Good friends with the textbook. Well, it's better than maybe some kind of other friends that you could make, right? Confident? Nervous? Nervous, okay, okay. First exam usually causes a little anxiety. So the first exam is usually the one exam of the year where, and of both terms actually, where time is a little bit of a factor. So I'm kind of wrestling with that as I'm writing the exam right now. And I try to make it as short as I can so that time's not a factor. But the shorter I make it, then the more points each question has. And so that's always a difficult balance, you know. So I try to get it down, but I find that that's sometimes a concern. And in this exam, you'll find that there's quite a wide variation in how much time people actually take on the exam. And so that's another thing. I think people work through different kinds of problems with very different speeds sometimes. So that's a bit of a consideration. I will tell you a little bit about my philosophy of writing exams. And my philosophy is that when I can, I want to cover the material as evenly and as broadly as I can, because that gives you as much opportunity to demonstrate your knowledge. If it's not covered broadly, then, you know, well, I studied this, and I studied for hours and hours, and it didn't even show up on the exam sort of thing. And hopefully that's not going to happen to anybody. I don't think it will. But that's just sort of some thinking about the exam. I don't try to trick anybody. That's not an aim. I really believe in trying to find your knowledge. And I will also tell you something I had a student who took my exam on the eCampus today. So eCampus students have a slightly different schedule than the classroom students. And she was, you know, she did pretty well, but she didn't do as well as she thought she should do, et cetera. And what I told her, and I'll tell you is the same thing, is that much as I would like for every exam to be a perfect measure of your knowledge, I don't think there is such a thing. So I told her, you know, as I said, she did pretty well. So it wasn't like she did badly, but she wanted to make a perfect score, and she did make a perfect score. And so I said to her, you know, you're upset because you made this, and you feel like it wasn't a good representation of what you knew. And she said yes. And I said, well, what if you had made 100 on the exam, and you felt like, wow, I really didn't feel like I knew that much. Would you be upset? And she goes, duh, you know, of course not. And I said, why not? Why not? Okay? Because the aim is the same in both cases. You want the exam to be a good measure of your performance. And I mention it to her because sometimes, I'm sure you've had this happen, you do better on an exam than you thought you were going to do, right? And so sometimes you don't do quite as well as you thought you were going to do, and hopefully, in the end, they all balance out. Okay? So I'm not trying to prepare you for the worst, so don't worry about that. The other thing I will say is, remember with exams that this class is graded on the curve. So because it's graded on the curve, you know, what matters is how you perform relative to everybody else. That's what a curve is all about. So if you walked out feeling like, oh my God, I bombed the exam, well, if everybody bombed the exam, then you're not quite as bad off as you thought. No, seriously, you know. So averages on my exams vary a little bit, but they're usually in the 60 to 70 range. If they get better than that, sometimes. They get worse than that on occasion, not very often. So they're almost always somewhere in the 60 to 70 range, which I think is really not that different from most college exams. So hopefully that's where you guys will all be. All right, so the way this works is I'm here to answer your questions. I won't bring up any new material, at least no new material that you're responsible for. Sometimes when I get babbling, I get talking about something I didn't talk about in class, but you won't be responsible if I talk about any new material in this session. So we'll start with questions. Who has questions? And by the way, when you ask questions, please, you know, be specific. You know, can you work through that again? is, doesn't really tell me what you don't know. And, that's what the videos are for, if it's just going over something again. So if there's something I can answer for you that you need clarity on, those are the best types of questions for me to answer. Yes?
I have a question regarding the enzymes and chemicals responsible for cleaving peptide bonds.
Yes?
Specifically, well, I guess all of them have a brief discussion of them in the United States, the carboxypeptidase.
Okay. So the question, I'm going to repeat the question so people who are watching on video will have a chance to hear them. So the question had to do with the agents that we use to cleave peptides. And specifically, he's interested in carboxypeptidases and what's the deal with those. So that's, if I can take liberties with what your question was, is what the question's about. Is that correct? Okay. So the reagents for cleaving polypeptides are right here on the screen. And you can see that I'm just burning myself with a laser. They're right here. Now, first of all, the reagents that you see here, I only listed certain ones that you needed to know. And so you should refer to the videos for that. The reagents that you see here are listed because they're specific, with one exception, that's the carboxypeptidase. They're specific in that they consistently make a break at a specific point within a polypeptide chain. So if we look at cyanogen bromide, it makes a break on the carboxyl side of methionine residues. Trypsin makes a break on the carboxyl side of lysine arginine. Thrombin breaks on the carboxyl side of arginine, et cetera, et cetera. The one that's not specific is carboxypeptidase. And carboxypeptidase I list because it's actually used in some very old techniques, which you're not responsible for, but it's actually used in some old techniques for determining the sequence of a polypeptide chain. Okay? So if you cleave off, so if you treat a polypeptide with carboxypeptidase, what it will do is it'll start at the carboxyl end and cleave bonds inwards. So to cleave off the amino acid at the carboxyl end, the only thing that attaches the amino acid at the carboxyl end is the peptide bond it has on the amino end. And so it cleaves on the amino side of the C-terminal, that is the carboxy-terminal amino acid. Okay? And it works sequentially. It'll just go inwards, 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 inwards. It does show some specificity, but to give you something very easy to remember, I say I'm not holding you responsible for the specificity. So you don't have to worry about that, not arginine, lysine, or proline. If I ask you what carboxypeptidase does, I expect that you're going to tell me that it cleaves on the amino side of carboxy-terminal amino acids in a polypeptide. Is that okay? Good. Yes?
So, would you be able to identify sequence, or at least how location of certain amino acids based on what sites were made off of polypeptide chain?
With carboxypeptidase or with any of them?
With any one of these.
Well,
what is the purpose of it?
okay, so what's the purpose here? All right. So the reason that I'm telling you about these reagents is all of them are useful for taking a polypeptide chain and breaking it into smaller pieces. Okay? Breaking it into smaller pieces. Each of those pieces can be analyzed more readily than can a long polypeptide chain. So the example I gave in class was that of 2D gel electrophoresis. And if I had a spot on that 2D gel electrophoresis and I wanted to know what protein that was, I would excise that spot and I would treat it with one or two of these reagents. So let's say I treated it with trypsin. Okay? Every place there's a lysine or arginine in that protein, that polypeptide chain, it's going to make a break. So I could analyze what's the molecular weight of each of these pieces that I got out of this. And using the techniques that I talked about in class, where the computer could go through and say, what group of amino acids, when joined together, would give a weight of 791.32, I could say, okay, this fragment has the following amino acids in it. Okay? if I was very careful and I looked at the breakdown pattern of that, the difference between the molecular weights of the fragments of a given peptide, I could actually see the sequence of those amino acids because the difference in corresponding pieces of the fragment will correspond to amino acids each of a unique molecular weight. So with careful analysis, I could take one of those fragments and determine the amino acid sequence of that fragment. Okay? I could do the second thing with a fragment over here. And I could do the same thing with a fragment over here. Now the problem is, I don't know if fragment A is next to fragment C, fragment B, fragment G. I don't know how to put the fragments together. Understand? All I know is I know the sequence of a fragment, a fragment, a fragment, a fragment, but I don't know how they come together. So if I wanted to know that, I would go back, get more of that same spot, and I would cut it with something that cuts, let's say, on the carboxyl side of methionines. Those aren't going to be the same size fragments. But some of those are going to overlap. In fact, each of those is going to overlap with the fragments that I got. So I can look and say, oh, this spans this break. So A must be next to F. F must be next to H, right? And then I can put the pieces together. So that's why we want to have multiple different specificities for cutting. And the fact that they cut at a specific place is very helpful because now I have specific sizes of fragments that I can analyze. Does that answer your question?
Yes.
Was it more than you wanted to know? Yes.
No, it's actually very interesting.
That's okay, I'm joking. But okay. What else can I tell you guys? Yes? What's the difference between a reverse turn and a bend? For our purposes, they're the same thing. Okay? They're the same thing. There's a lot of terminology with respect to protein structure that we don't go into. And the reason we don't is this is not a biophysics class. So for our purposes, if you understand the importance of bends, okay, I'm happy. The importance of bends is that they're really what ultimately orients the secondary structures within the tertiary structure. What does that mean? It means that if I have an alpha helix and I have a bend and then I have an alpha helix, right? The bend has determined the relationship between the position of the top alpha helix and the bottom alpha helix. Well, when I bring in the bottom alpha helix, I'm now talking about tertiary structure, whereas the alpha helix itself was secondary structure. So the bend is orienting the secondary structures within a larger tertiary structure. Does that make sense? Yes?
Primary direct interactions is just between the amino acids that are directly adjacent to the amino acid we're talking about. Secondary is less than 10 away, and then tertiary is more than 10, and then quaternary is the same as polypeptides in the same group.
Correct. So what she just said was that a primary structure, I'm going to change your primary structure just a little bit. Primary structure actually refers to the sequence, and sequence ultimately is one next to the next, next to the next, to the next, to the next. But yes, primary structure is sequence. Secondary structure are interactions closer than 10 amino acids apart. Tertiary structure are interactions between amino acids more than 10 apart. And quaternary structure are interactions between individual polypeptide chains. Yep. And we do see, we'll talk about this actually, I keep saying we'll talk about it, but I'm not teaching 451 next term, so I won't be talking about it with you, but somebody else will be talking with you about it. With respect to RNAs, when they fold up, can also have tertiary structures. And we don't really think about them with quaternary structure, but they do actually have tertiary structures that they can fold with as well. So they're kind of analogous to polypeptides in that sense. Should we go home? Can I go home, Mom? Yeah, sure.
Can you go over the 5
Okay. The Ramachandran plots? Okay. So Ramachandran plots, I hope don't concern you too much, because they're really there to help you to understand something. And I'm not going to make you draw a Ramachandran plot, as I said. So, but that's fine. I'm happy to answer your question. So let me go through and answer your question and hopefully put you at ease a little bit. So let's go back to what made this up in the first place. Okay. So if we have a polypeptide chain, okay, I pointed out that peptide bonds have characteristics of double bonds because of resonance. Okay? And so if we look at peptide bonds, there's a peptide bond right there, and there's a peptide bond right there. Okay? And peptide bonds cause lack of rotation to occur. And with lack of rotation, and what you remember about peptide bonds from your freshman chemistry, or at least double bonds from your freshman chemistry, or your organic chemistry, rather, you realize that wherever there's a double bond, you have a flat structure. And so I like to think of it as a plane. So I can think of this peptide bond over here being a flat structure, and I can think of that peptide bond over there being a flat structure. My thumb corresponds to the bonds that are to this alpha carbon right here, and the alpha carbon is my nose. All right? So I always use that analogy because it sort of helps to orient everything. These guys, because this bond and this bond is each single bond, can rotate. Single bonds can always rotate. Well, even though it can rotate, it doesn't mean it can rotate through a full 360 degrees, because these things that are sticking out here, and there are some sticking out down here as well, can't show you accurately, but these things that are sticking out are bulky, and they place constraints spatially on how close together things can be put. So polypeptides have to obey the laws of nature. They can't put two nuclei too close together, or you make a fusion reaction, which of course releases a lot of energy, and of course we don't do that. So because we're dealing with standard chemistry, all right, they have to orient themselves in space so that they don't go through those limits. All right? So they will twist themselves into the stablest structure that they can. And this twisting is occurring across these bonds that are on either side of my nose. Notice that the plane, that is the double bond, which is my hand, is flat, but that the single bonds are rotating. Okay? So Ramachandran said, okay, if they can rotate, there must be a stablest structure that they can have. Can we predict that? And that's what gave rise then to the Ramachandran plot. The Ramachandran plot was originally purely theoretical, based on what we knew about energies and closeness of nuclei. So what he found when he did the analysis was that there were three main regions that occurred that had stability, and the two primary ones were right here. Okay? The two primary ones were right here. The guys right here, this one and this one, refer to different kinds of alpha helix. Okay? Alpha helices, of course, these angles give rise to alpha helices. These kinds of structures were predicted by Linus Pauling, and this is a nice verification of that they're chemically stable. And you see that this is plotted as 360 degrees of rotation of the phi angle, the psi angle, and 360 degrees rotation of the phi angle here. So we have 360 and 360. Which ones are the stablest? This big structure up here is what gives rise to the beta strands. This is the folds that I showed in class. So, well, why do we have to know Ramachandran plots? Well, why do you have to know anything, right? You need to know Ramachandran plots because Ramachandran plots help us to begin to visualize protein structure. Protein structure is, I hope you begin to walk away with, is extraordinarily complicated. It's extraordinarily complicated. The Levinthal's paradox that I told you about about how long it would take a structure randomly to assume its 3D configuration tells you all of the possible ways that something could come together. And it tells you that there's an extraordinary amount of complexity within that protein structure. This is a simplifying mechanism. If I say that there's only certain angles that give rise to stability, then when I go to predict structure, I can throw out all the structures that have the wrong angle. Because most likely, they're not going to give rise to a stable structure. That's the value of a Ramachandran plot. Yes?
And so would these plots vary depending on the R group that's on?
That's a very good question. Will these plots vary according to the R group that's on there? And the answer is they will. These are typically done, the predictions are typically done with a given R group, okay, in multiple forms. So you could imagine that, and this is typically done with something like alanine or something fairly simple. What if I did such a plot with tryptophan? Okay? Might I see some constraints and some other angles that would be slightly different? And the answer is yes, you very well may. Okay? You don't have to worry about that. This is just to understand the basics, that there are limitations in terms of stability for these angles. And so we can think of the structure of a protein at a very close level in very simple terms. If I'm talking about two amino acids joined together, okay, I could say angle, angle, and I've got it, right? And then I think the next one is angle, angle, and I've got it. Angle, 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 angle. So at a close range, I can tell protein structure in very simple terms. In a longer range, where I've got bends and other things that come into play, it becomes much more complicated to consider. But at a short distance for very small things, the Ramachandran plots actually help me to better understand protein structure. You're not going to draw one. You're not going to have to tell me which angle is phi, which angle is psi. That's just memorization for no purpose. Okay? But I think you should know that a phi and psi angle refers to a rotational angle around that alpha carbon. I think that's an important thing. Yeah. Question? Okay.
Proximal and distal histidines and the role they play in hemoglobin and myoglobin.
Okay.
How that pertains to oxygen.
Okay, so proximal and distal hemoglobins are something that I haven't talked about in class. So rather than bring it up here and get a lot of people worried about it, I won't talk about that here. If you want to come talk to me, I'll be happy to talk to you. But there are two histidines that play roles in the heme structure of hemoglobin. One holds onto the iron atom, and the other one is above the plane and actually interferes with the binding of other substances there. That's what you're talking about, but I'm not going to go into it here. Plus, that takes more time anyway. So, but come see me. I'll be happy to try to talk about that. Or not try to actually talk to you about that. I have no trouble with talking. It's just thinking every now and then gets in my way. So, okay, other questions? Yes, Deirdre.
So, kind of related to hemoglobin. This is more just like conceptual.
Okay.
So I know that oxygen is more likely to release at a lower pH. So then, like if you were in a slightly basic solution, the hemoglobin would be more willing to hold on to that oxygen.
Correct. So our question was, the Bohr effect says that the more acidic the solution, the more oxygen hemoglobin releases. Conversely, the higher the pH, the more the hemoglobin holds on. There are limits. Well, now we've got to know about limits, too. No, not really. You know there are limits to the stability of a protein structure based on the pH of the solution it's in because of folding, right? So if you change the pH too much, either too high or too low, you will denature the protein, in which case, nothing's going to work. Was that your question, or was there more than that?
Oh, yeah, no.
That was it? Okay, that was easy. I like easy ones. I know the answers to those. Yes, sir.
I guess an overview of its role again, and what needs to happen environmentally to cause hemoglobin or myoglobin to let go of that.
Okay, good question. So 2,3-BPG, a little bit of an overview on 2,3-BPG, and what causes myoglobin or hemoglobin to let go of it. First of all, myoglobin doesn't bind 2,3-BPG because it doesn't have a donut, right? So only hemoglobin. That'd be a good test question, right? Where on the myoglobin molecule does 2,3-BPG bind? I hear this scream in the back of the room, right? Ah! I don't know what to do, right? Okay. So 2,3-BPG. So I'm going to tell you a little bit more then I told you in class, you won't be responsible for it because it helps to understand this a little better, okay? So 2,3-BPG is a byproduct of the breakdown of glucose. I think I mentioned that in class. It's a byproduct. It's not a main product. It turns out it's a relatively trivial byproduct of the breakdown of glucose. Okay? The more breakdown of glucose you have going on, the more of this molecule you release. I probably said that in class too. So the concentration of 2,3-BPG is a measure of the concentration, is a measure of the metabolism of the cell that produced it. Rapidly metabolizing cells will produce more 2,3-BPG than cells that don't metabolize fast. So a muscle cell that's exercising is producing a lot of 2,3-BPG. A skin cell that's on my thumb probably isn't producing very much 2,3-BPG. Okay? Now, I haven't gotten to the part I didn't tell you in class, but I'll save that for a minute from now. Because there's this very nice signal, as hemoglobin is passing through the body, when it encounters 2,3-BPG or it encounters rapidly metabolizing cells, it gets the signal to let go of oxygen. Because rapidly metabolizing cells need oxygen the most because they're burning the most energy, and energy is necessary for efficient energy production. Oxygen is necessary for efficient energy production. I gave the example in class. If cells have oxygen, when they burn glucose, they get about 38 ATPs. The same cell burning glucose without oxygen gets 2 ATPs. Okay? So a cell that is a muscle cell that is rapidly needing energy, and of course muscle cells are flexing because of energy. They're flexing, they're burning ATP to make this happen. They need to produce ATP. Okay? Cells that are rapidly metabolizing have a greater need for ATP, and more ATP is going to come from burning glucose in the presence of oxygen. Okay? All right. Now, so that signal is very good. There are 3 ways I talked about in class. There are 3 signals. Remember what they are? 2,3-BPG. Say what? Protons. Carbon dioxide. Okay. Now, she said lactic acid. Lactic acid is actually a byproduct. It's not a signal of the rapid metabolism. But lactic acid will be produced when oxygen is absent. Okay? You don't need to know that. I haven't talked about that. Okay. All right. Everybody just goes, it's gone, right? I just cleaned my brain out, right? I got room for something else now, right? Okay. Now, the 2,3-BPG story is interesting because, first of all, his question was, how does it get off of the hemoglobin? And the way it gets off of the hemoglobin is by virtue of the fact that it's not a covalent binding in the first place. It's not a covalent binding. So things that are not covalently bound, we will see come off. They come off, and oftentimes they just rebind. They come off, they go on. That's why I always talk about this coming off, going on, coming off, going on, coming off. Because the most likely thing they're going to do is just come right back together. You go off a little ways and you come back together. Okay? So these aren't things like we think of, again, in the real world, where you grab a hold of something and as long as I squeeze my fist, it's there. Okay? That's not the analogy. The analogy is it's sort of doing this. Okay? That's what's happening in a non-covalent binding of molecules. Now, I tell you that because when 2,3-BPG binds to glucose, it's in this sort of state, this sort of quasi-bouncing back and forth state. And as hemoglobin is making its way back from the tissue to the lungs to get more oxygen, along the way, some of the things coming off, instead of going back together with hemoglobin, come off and are grabbed by a cell, which a cell uses for energy. Okay? So that's how the 2,3-BPG gets removed from the hemoglobin. And it's purely due to the fact that it's not a covalent binding that's holding that together. If it were covalent, then it would be on there permanently. You would use hemoglobin once, and that would be it. You wouldn't use it much again at all. Yes, Deidre.
So the 2,3-BPG changes the structure of the hemoglobin?
Correct. So 2,3-BPG converts hemoglobin from the R state into the T state. The T state is the low oxygen affinity, and that's why the oxygen is released. Okay? Now, it's important that 2,3-BPG come off before it gets back to the lungs, because as long as 2,3-BPG is there in this state, okay, the hemoglobin is in the T state. So if hemoglobin, and it's locked in the T state. It's not going to flip out of that T state until the 2,3-BPG is completely gone. So if the 2,3-BPG is not completely gone, when it gets back to the lungs, hemoglobin is going to remain locked in the low efficiency of binding oxygen state. The hemoglobin is going to get relatively little oxygen, and that's why the smoker has low oxygen concentrations in their blood, because they've got a lot of 2,3-BPG, and their hemoglobin is coming back to the lungs like that. The hemoglobin is more frequently in the T state, consequently. Now, the thing I didn't tell you in class, but that several people have asked me about, so I'm going to mention it here, but you're not responsible for it. We'll talk about it when we talk about glycolysis and metabolism. Is, well, why is the 2,3-BPG higher in the blood of a smoker? There's really two reasons, one of which is a catch-22. One of the reasons is very easy to understand. Cigarette smoke is full of carbon monoxide. Carbon monoxide competes with oxygen for binding, and carbon monoxide, by its very presence, reduces the oxygen concentration. Okay? 2,3-BPG, you've seen, reduces the oxygen concentration. Okay? So why is 2,3-BPG there? It's there because the smoker has less oxygen, and that less oxygen makes for less oxygen. So the less oxygen a cell has, the more glucose it has to burn to get the same amount of ATP. So this thumb cell, this skin cell on my thumb that I was talking about before, in a smoker, okay, is going to be getting less oxygen. The muscle cells are going to be getting less oxygen. And those cells still need ATP. How much more glucose do they have to burn in the absence of oxygen than they do in the presence of oxygen? Well, in the complete absence, they need to burn 19 times as much glucose, because they get 2 versus 38. That's 19 times. So if you reduce the oxygen concentration in the blood of a smoker by any means, carbon monoxide or whatever, what happens to the metabolism of that cell, or any cells? It's going to go up. And the more it goes up, the more 2,3-BPG you make, and you got a vicious cycle. Somebody said that when I talked to them about it yesterday, they said it's a vicious cycle. I said it's exactly what it is. Because the lack of oxygen makes for more lack of oxygen. That's the cycle that a smoker is in. And that's why when smokers quit smoking, they can frequently discover a fairly noticeable difference fairly quickly. So that's what's up. Yes?
I was just going to kind of go along with that. So does that mean it's like a weight loss tool? If they have to burn more glucose in order to...
So it's a good question. Is smoking a weight loss tool? It's not a silly question, actually. A lot of people find two things when they quit smoking, both of which lead to increased weight. Okay? One is that food tastes better. And so that happens. And yeah, and because they start using their oxygen and making ATP more efficiently, as they're getting more ATP out of everything they eat, then they've got all this extra energy that's going to be made into fatty acids. So it's not at all uncommon for people to gain weight when they quit smoking. Yeah. Yes?
So is this something that happens if you smoke anything, or is this a cigarette?
You're the second person to ask me that question. Okay. I can answer this question because we are legalizing marijuana in this state. Okay. So I can't give you a definitive answer, okay, because I don't know the definitive answer. There are some things of smoking marijuana that will parallel smoking, one of which is the production of reactive oxygen species. We haven't talked about these. We'll talk about them later. When you inhale anything that's being burned, you're getting some reactive oxygen species. So that will probably mimic some of the things of smoking. The second thing that happens with the inhaling of things that are burned is probably you're getting more carbon monoxide than you would otherwise. So that may have some of the effects that you see in smoking. Marijuana, of course, doesn't have tar and nicotine, which are the addicting substances in cigarettes, why marijuana is not addictive. So my advice is don't smoke marijuana. Put it into brownies, guys. I mean, think about this, right? It's a no-brainer. That is not an official thing. That is a joke. But anyway. Anyway, so what other questions? I've heard the DEA on me now. As I got to get Ahern here, right? That's not my medical advice. By the way, I can't give medical advice, so that's a disclaimer. Okay.
Okay, so you mentioned that when the hemoglobin is T3BP, it needs to release that before it gets into the lungs, or else it can't have
Yes.
oxygen. What causes T3BP to be released?
Okay, so again, what causes 2,3-BPG to be released? The fact that it's not covalently bound. It's solely that. So a non-covalent bond is literally, like I said, I mean, it's like this. It's not something that just rigid and stays here. Okay? I mean, we talk about it being, present in a certain state, locking hemoglobin in a certain state, but it's really not permanently there. And that's the best answer I can give you for that. So, yes.
So you say that when it comes off, most times it is eaten by a cell.
Yes. It is usually eaten by a cell. That's right.
So I guess is there more like the need for this T3BP to be like taken by the cell? Like
Yeah.
if it's floating between the cell wants to eat it, and hemoglobin is going to tend to this way. The cell grab it really.
So there's a fair amount of affinity for virtually anything that cells can grab onto and get energy. So this is true whether it's glucose, this is true of metabolites, this is true of amino acids, et cetera, et cetera. So yeah, there's no shortage of desire on the part of the cell to grab a hold of that. You don't get much energy out of it. Okay, so it's like I mentioned in class. It's not like that the cell makes up for the loss of efficiency of glucose by grabbing 2,3-BPG. Because if that cell that grabs 2,3-BPG still has low oxygen, it's going to get one additional ATP out of it, is all it's going to get. So it's not going to make up a big deficit. Yeah. Yes?
So in isoelectric focusing, there's a pH gradient needed.
Yes?
How is that done? Can you do
Okay, so the question is, in isoelectric focusing, there's a pH gradient that's made. How is that gradient made? Okay. So it's actually kind of a cool trick. So go back to this guy and isoelectric focusing. So it literally occurs by the way that I mentioned to you, which is that you have this tube that has these little beads, they're little gel-like beads that are in there, that are coated with varying levels of charge. That's really what is there. So that when you start out with the tube, and it's gelatinous, so it's very sort of fluid in the first place. They're not beads like we use in gel exclusion chromatography, for example. They're not rigid beads, but that they are sort of a gelatinous mix that's there. When you take those beads that have varying charges on them, and you have a whole spectrum of charges, when you apply an electrical field to it, those that have the most negative will be pulled the closest to the positive electrode. Those that have the most positive will be pulled closest to the negative electrode, and then a whole gradient of those in between. That gradient of charge that you have there is a pH gradient. That's what it is. And so in the middle of that overall thing, you will have things that have no charge. I keep doing that. Have no charge whatsoever, and that'll be inside of here. But the proteins sort themselves according to that gradient. If you want to think about it as a gradient of charge, that's perfectly fine. You don't have to think of it as a pH gradient.
apply voltage and then you take the voltage off and then add the protein?
Okay, the question is, when do you add the protein? Actually, no, you don't. So what you do is you take this slurry of material, you put your proteins with the slurry of material, then you apply the voltage. So the proteins move with the charges. It's a good question. And you could actually do it that way. You could apply the proteins to it, But it'd be, why would you do it after the fact if you can sort them both at the same time? If you apply the proteins to it after it's set up, I think it should separate just the same way. I don't run these, so I can't tell you, but I think it should separate just the same way. Okay? Yes, ma'am.
For PI, in order to calculate that,
Yes.
we would either, if given a hypothetical reaction, we'd be given the charge, or we would have to know if given an amine what the charge was to be able, so we know where zero is.
So her question is, in calculating a PI, basically, I'll put it in broader terms, what would I need to know to calculate that? Would that be fair to say?
Would it be to know the amine, or you'd be given the charge?
So in order to calculate the PI of a compound, you would have to be able to determine what the charge of it was with all the protons off and with all the protons on. And that's sort of stepwise going through there. That you would have to know that. If you didn't know that, then there wouldn't be any way for you to determine where the PI of the molecule actually is.
You should have to, it is all visual. When I'm sitting there counting them up, that's what's going to happen when you're looking at the chart.
You're going to need to know that. That's correct. Yeah. Okay? Yeah. Back here, Stephanie. How I what? How I group the amino acids. Okay. So I've somehow got to get all the groupings consistent, and everybody worries about this. So the groupings of amino acids that you should be concerned with are actually shown right here. There's your groupings. You know those groupings, you're in good shape. Okay? So that's the level of detail necessary for knowing the groupings. You should also know if they ionize, how they ionize on their R groups. Okay? That's the only other piece of information, as far as I'm concerned, you need to know. So I'm trying to spare you having to memorize structures. Okay? Question back there. Can you use mercaptoethanol and DTT interchangeably? Since I haven't talked about DTT, I guess if I used them interchangeably, you guys would cut my arm off. So, but they are equivalent in terms of their function. But I'm only going to give you things that I have talked about. Yes? Do you have to know pKa's of R groups? You will not need to know pKa's of R groups. You might, I mean, it's possible you might have to calculate one, but that would be only if you had enough information to do it, right? Yes?
So I noticed, at least in my checks here, that you're referring to the 19 it was broken. It went to the same as the...
My link here is broken. Is that what you're saying?
Yeah.
Oh, sorry about that. Yeah. Sorry about that. Okay, I'll fix that. But you don't need to know the structures anyway, so it shouldn't be a problem. But if I fix it now, you become responsible for knowing the structures. Is that right? You can kill her. It wasn't my idea. It was her idea. Yeah. Glutamine and glutamate are not the same thing. No. So glutamine is glutamate refers to glutamic acid. Okay, glutamine refers to the carboxyamide version of that. So here's glutamine right here, and here is glutamate right here. They're not the same thing. Okay, so I think the structures hopefully should be here. So if you look at glutamine, all right, glutamine has instead of an OH on its R group carboxylate, it has an NH2. All right. If you look at glutamate, it has on its R group carboxylate, it has an OH instead of an amine. So glutamine is it's called the carboxamide equivalent of glutamate. Asparagine is the carboxamide equivalent of aspartic acid. You don't need to worry about that, but they are different things. Okay, yes?
I was wondering if you could go over what we talked about the velocity of amino acids and
The velocity of amino acids, I'm not sure.
some of the stuff we went over on enzymes and
So the velocity of a reaction you're talking about?
some of the stuff with like the Metabolic mentioned constant, some of that. Still trying to
Okay, anything specific?
I guess in terms of being able to look at that and take them in values and put
All right. Well, let's go to Km, since you've asked about that one, and talk about that briefly. Okay. So, let me, as I've done before, back up a little bit and give some background, which hopefully will help you to understand this better. So, I think it was you and I who spoke today, but if it wasn't, it was somebody else that I spoke to. And they had some real concerns about VO versus Vmax. Was that you or was that somebody else? Somebody else? Okay. All right. So VO is initial velocity. And they were very concerned because when I showed this plot, they said, is this VO or is this Vmax? And then the question was, well, what's the difference between VO and Vmax? So let me first of all tell you what VO is. Okay? If you remember, I said when we measure an enzymatic reaction, we want to know how fast it goes. And if we let the reaction go for too long, we get no velocity because it's at equilibrium, and at equilibrium, there's no movement because forwards equals backwards. We can't measure because we can't see the formation of product because the product is already there. Okay? So to measure the velocity of a reaction, we measure its velocity very quickly, okay? And we call that quick measurement initial velocity. And it's called initial velocity because we're measuring the velocity before the product begins to accumulate. Because once the product begins to accumulate, then the reverse reaction can start happening, and that's going to throw off my calculations. Right? Okay, so VO is the measurement of the velocity, all right, before the product begins to accumulate. Now, this was what surprised the student that I talked to. Vmax is a VO. They're all VOs. Every velocity we measure is an initial velocity. Okay? Now, Vmax technically is a theoretical velocity. We can estimate it. It's technically the velocity at infinite amounts of substrate because at infinite amounts of substrate, we've saturated the enzyme, right? And Vmax is also occurring before there's any product that's there, so it's the absolute initial velocity, right? That's really what Vmax is. But it's an initial velocity. Every point that we did here was an initial velocity. In fact, every single velocity measure we do in kinetics is an initial velocity. So don't let VO throw you off. I just write it as V because it's a velocity and we do them all the same way. Make sense? Now, that was the first thing. Okay? Now, you asked not about velocity, but you asked about Km. So, like I get off on a tangent, right? All right. So once I've established what Vmax is, I'm able to determine Km. And Km, as I said, is the measure of the enzyme's affinity for its substrate. The substrate, of course, is the molecule that it binds to that it catalyzes the reaction on. Right? Enzymes vary a lot in their affinity for their substrate. Okay? Two parameters we've talked about with enzymes that vary a lot. One is their affinity for their substrate, and the other is their kcat. Remember that Vmax is not a constant for an enzyme. It depends on the amount of enzyme that I have. So I can't say the Vmax for this enzyme is blah, blah, blah and have any meaning unless I know how much enzyme is there. Now, getting back to Km, I keep getting off the subject. Okay. The way that I determine Km is by first determining the Vmax. If I don't know the Vmax, I can't determine Km. I first determine the Vmax, and then I go to Vmax over 2 and ask what substrate concentration gave that Vmax over 2. And the substrate concentration that gives Vmax over 2 is the Km. So Km is a substrate concentration. It's a specific, excuse me, it's a specific substrate concentration. And substrate Km values that are low indicate high affinity, meaning it doesn't take very much substrate to get the reaction to half Vmax. The enzyme is grabbing hold of it quickly and catalyzing a reaction on it. If I have a high Km, the enzyme has a low affinity, meaning that it takes an awful lot of the substrate to get the enzyme going, basically. Does that help? Okay. Now, Km, people say, well, a couple people have asked me, well, why Vmax over 2? Why not Vmax over 3 or Vmax over 4? Okay. And it's actually a good question. The Vmax over 2 arises from a mathematical derivation, and we don't do the mathematical derivation here. We could do that, but that would probably take about a week, you know, and I don't think you want to take a week to do it. But the Km over 2 is not just picked out of the air. There actually is a reason why that value tells us the affinity for of the enzyme for the substrate. Okay? Good questions. Yes? An example of how the R groups on the amino acids ionize? Okay. So if I were to go to, let's say, one of the ones that ionize, let's say lysine. Okay. Oop. I'm sorry? Aspartate? Okay. That's fine. So let's go to aspartate. Actually, maybe for that, I almost need to write it on the board. Let me see. Okay, hold on. So if I if I turn it off, it's going to take 5 minutes to start back up again. I can use the dot can. You're right. You are absolutely right. And if somebody has a piece of paper, I can write on a piece of paper. What's that? Oh, it doesn't matter. You get an A on the exam, by the way. So, damn! I wish I'd given him that paper first, right? So, okay, dot cam is not coming on, guys. There we go. Okay. If we get it oriented the right way. All right. So let's see if my handwriting stays on it. Okay, so you want to see aspartate. Here's what aspartate looks like in its various forms. Okay, so there is aspartic acid's R group. I'm writing on the top. Write it consistent. All right, and then NH3+. Now, this aspartic acid that I just drawn has all the protons on. Protons on the carboxyl on the R group. It's on the carboxyl on the alpha carboxyl. It's also on the alpha amine. The overall charge of this guy would be +1. Okay? Now, if I told you that the pKa values for this guy were alpha carboxyl equals 2.2, alpha amine equals 9, and alpha R group carboxyl equals 4.4, then that tells me that if I started with all the protons on and I started raising the pH, the first proton that would come off would be the one that corresponded to the 2.2. And so that would go to COOH, CH2, C, and that's an H here, CO minus, and NH3+. Because that's the alpha carboxyl. Pull that down. The alpha carboxyl has ionized. This is the alpha carboxyl right there, and we can see that it's ionized. Okay? The overall charge of this guy would be 0, when I ionize or keep raising the pH, the next one that's going to come off is the R group carboxyl of 4.4. And so when that comes off, it's going to look like this. Okay, and so this is going to have a charge of -1, -2, +1, or an overall charge of -1. And then the last one to come off is going to be the alpha amine. And when I do that one, what I get is something that looks like this. And now I've got an NH2, which has a charge of 0. And the overall charge of that guy would then be -2. Yeah?
We wouldn't be able to do this without the values you gave us?
Well, you would need to know you're talking about the pKa values? Yeah, right. So you wouldn't need to know, you wouldn't need to know those pKa values, but I could give you those pKa values, yeah.
So you said earlier that you would need to know that if they ionize, how they ionize.
Yes.
So wouldn't that mean that every
every amino acid has an alpha carboxyl and an alpha amine, but not every amino acid has an R group, right? So if we were talking about a polypeptide chain, the amino acids in the middle don't have an alpha carboxyl and alpha amino because they're in a peptide bond. And so if they don't have an R group, then you don't know, they actually wouldn't ionize. Does that make sense? So for all practical purposes, you need to know which ones have R groups that ionize, because every one will have alpha carboxyls and alpha amines that ionize. Does that make sense? Question here, yeah.
So the PI for this one will be at 2.2?
Well, the PI, all right, so you're guessing here, so don't guess, all right? Let's draw it out. So if we look at the titration plot for this, where we plot pH, can you see all that? Okay, I can't see what shows up, versus OH added, all right? So we see it rises to the first pKa value, flattens, rises to the second, flattens, rises to the third, flattens, and then takes off, right? So the first one corresponds to 2.2, the second one corresponds to 4.4, and the third one corresponds to 9. So what I strongly recommend you do is what I've just done right here, which is draw it out. That way you won't be guessing. What's the charge on the molecule with all the protons on? Well, we decided it was +1, and that was right here at the bottom. The charge right here, where the first proton is completely off, is 0. The charge here, where the proton is completely off, is -1. And the charge here, where the proton is completely off, is -2. So if you were to determine the PI, it would be the average of the two pKa values on either side of the place where it's 0. So it would be equal to 2.2 plus 4.4 all over 2, and that turns out to be 3.3. Oops, there's my calculations down there. Okay? So don't guess. And PI values will never be pKa values. They will never be pKa values. Yes?
Do you want us to consider cysteine a polar amino acid or an acidic one?
Okay, so I grouped it as a polar amino acid, but you should know that it ionizes to make an S-. Okay? Yes?
So if you had a polypeptide that was just glycine,
Polypeptide was just glycine?
it was just glycine,
Okay.
a bunch of them in a row,
Uh-huh.
to calculate the overall charge, you could just take the alpha amine and
Yeah, okay. So our question was, if I had a polypeptide and it was polyglycine. Glycine, 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 glycine. I like to say that. It's kind of fun, okay? All right. If I was calculating the overall charge, first of all, glycine has no R groups that ionize. I would simply need to know the charge of the alpha amine and the alpha carboxyl, and I would have it. But that also leads us now to the way to calculate the charge on a polypeptide. When I look at a polypeptide, it's very simple. I need to know the charge on the alpha amine and the alpha carboxyl on any polypeptide, and then I need to know the charge of any of the R groups, and I'm done. So I might have 50 amino acids, but if there's only 3 that have R groups that ionize, Then I've got alpha carboxyl, alpha amine, 3 R groups. Bingo, I'm done. Make sense? So that way, if you know which R groups ionize, you're set to go. Question here. Oh, okay. I'm reading your mind. Yes, ma'am. Can we do something else? Yeah, let me ask if there's any other questions on this.
So we're asked for the charges at a specific pH. And we just look at that region between the two plateaus. And the pH falls between those regions, we can know that's the charge. So the difference between pH between 2.2 and 1.4, that's the charge be 0.
So remember so her question was, how do I predict the charge, basically, all right? So you have to use the rule that I gave you about the proton being on or off with the respect to the pH being lower than the pKa by more than one value, the proton is on. Higher than the pKa by more than one value, by more than one pH unit, the proton is off. Then use that information to make your determination as for the charge. Yes. But you can't say that between 2.2 and 4.4 it's 0, because at 2.2 I can't tell you the charge. Half of them are one charge, half of them is another charge, right? Yeah. Okay, other questions on this? Not trying to shut anybody up. Okay, you had another question then. What was it?
Just go over
MALDI-TOF. Okay, so MALDI-TOF, going back to my computer. Okay, so MALDI-TOF is in here. And where do I have MALDI-TOF? Oh, this was the one I was going to get you a figure, wasn't I? And I didn't have a figure. Okay, so I still haven't gotten you a figure. I'm going to have to talk you through it. Okay, let me try to talk you through it. How can I help you with respect to MALDI-TOF that doesn't use a figure?
Just you write in your highlight it says by measuring the time it takes the
Okay.
charge molecule to go to the detector.
Okay.
Once the charge massive, that heavier molecules will have a lower charge than that region, and lighter molecules that travel slower.
Okay, you want some elaboration on that. Okay, so I apologize for not having a figure. So I know it's sometimes difficult to visualize this. But let's imagine I have right here on this table, I have a tube. Okay? And the tube has a vacuum, so there's no air in there. All right? At this end of the tube, there's a detector. And the only thing that it does is when an ion hits it, it gives a beep, it gives a blip. Okay? So this end is a detector. Ion hits it, it gets an electronic signal. Blip. Okay? This end over here has a place where I place my sample in. Now my sample is, let's imagine I've taken a peptide, I've chopped it up with one of the enzymes, so that I've got a fragment, and I want to determine what the molecular weight of that fragment is. And for simplicity's sake, we're going to say there's only one in here. We can actually, it's a more complicated way we can do more, but we're going to have one thing in here. All right? Detector tube, right? I put my sample in, and my sample goes on something that's like the head of a pin. And it's embedded in a little crystal-like structure. So when I put it, and the reason it's embedded in that is so when I put it in the vacuum chamber, it stays put and doesn't evaporate because it's a complete vacuum. All right? So I've got my sample sitting in here, and it's ready. I have a laser that can fire in here, and it's going to fire, and it's going to fire right onto that sample on that pinhead. Okay? When it does, the material that's in there vaporizes, meaning that it leaves the pinhead and goes into the vacuum of the chamber. As it does that, it gains a positive charge. You don't have to know how or why, but it gains a positive charge. That's necessary for this analysis. Okay? So now I've got a fragment in here that has a positive charge that I have instantaneously vaporized with a laser. That was time 0. Okay? Time 0 is when the laser hit the sample. Now, this guy's positively charged, and down here I've got this detector that has a negative charge. I've also got a positive charge back here. So just like in electrophoresis, the positive is going to repel the positive, and it's going to come this direction towards the negative. Right? So we all agree there's going to be a pull of this ion towards the detector, and it's going to hit the detector. Right? Okay. Let's imagine this sample, instead of having one thing, I had two things. Let's imagine I had one thing that had a molecular weight of 500 and one thing that had a molecular weight of 250. And they both had one positive charge. Okay? There's no barrier like there is in electrophoresis. There's a vacuum. There's nothing in between the two. There's simply an attraction and a repulsion that's driving it that direction. They both have the same charge. They both have the same attraction. And they both have the same repulsion. The only difference they have is one has more mass than the other one does. Well, like the analogy I gave in the class, if you're trying to push a Prius compared to pushing an SUV, you're going to find that pushing the SUV is going to be harder to do. It's going to take you longer to push the SUV the same distance as you push a Prius. And the 250 is going to move faster than the 500. In fact, the 250 is going to move exactly twice as fast. So we can determine then, by measuring the time it takes to go from the laser until we get a blip, if we measure that time very accurately and we calibrate our instrument, we know what the mass-to-charge ratio is. In this case, it's 500 to 1, or it's 250 to 1, depending upon how long it takes to hit that. Does that make sense?
Yeah, so when it gains a positive charge?
It gains a positive charge. So the question is, does everything you put in gain one positive charge? Some will gain 2, some will gain 3. But generally, to keep it simple, if you think of 1, you'll be on the right track.
So what if it's like 250 with a positive 3 charge and 500 with a 1?
Okay. You could do the math, right? You could say, all you can say is how long it takes and what the mass-to-charge ratio is. So if you had something that the example I gave in class was a 500 with 2, would travel exactly at the same rate as a 250 with 1. Because the mass-to-charge ratio in both cases would be 250 over 1. Make sense? Well, that's a more complicated analysis. But all I care about is that you understand the principle. I'm not going to make you do a computer analysis to determine if you have 2, 3, or 4 that's on there. Okay? Yes? On the okay, let me make sure. Any other questions on MALDI-TOF? Okay, go ahead. Okay, all right. So let's look at 2D gel. The left and right, negative, positive, are totally arbitrary. So left and right doesn't matter. All right. So in this case, the positive is over here, the negative is on the other side. But I could just flip that either way. It doesn't matter, right? Now, the largest will always be at the top. That's true for any of these because the second dimension is moving downwards, and that moving downwards in electrophoresis means that the smallest guys will move the fastest, going down this way. So this is going to be the smallest, this is going to be the largest, this is going to be the smallest, this is going to be the largest. So plus and minus just has to do with which way I lay the thing down. No, the one in the middle would be the most medium charged. Yeah, so the ones that would have the closest to 0 charge would be right here, not size, charge. If I said size, then that's not right. That's it?
Yeah.
Okay, yeah.
So in
So if you run any gel too long, what happens is your sample runs off the bottom. So yeah. So, well, there's a couple ways of doing it. One, you run a lot of these, you have an idea how long it takes. But a lot of times people will put a dye in there that has a known size, and they can just follow how far the dye goes and say, okay, the dye's at this point, then my smallest proteins are at the bottom. So, good questions, guys. Yeah.
Can you give an example of when you use Western blot?
When would I use a Western blot? Let's imagine I said to you that I have a mutant cell, and this mutant cell is cancerous, and I think that this mutant cell is not making as much of a particular protein as a normal cell. The question is that true? So I would take the proteins of the mutant cell, bust open the cell, take the proteins of the normal cell, bust open the cell, and run them on a gel, treat them with the antibody against that specific protein, and ask, do they both have it? If so, which one has more? Now you might say, well, why not do a 2D gel? The answer is a 2D gel is a hell of a lot of work. All right? So this tells you pretty quickly and easily if you have the thing that you're after.
So you need to know the identity of the protein. You have to know the identity of the protein.
You do. So you need to know the identity of the protein that you're looking for because you have to have an antibody against it. And to get that antibody, you would have to have originally isolated that protein, or somebody would have had to have done that.
So all Western blotting does is quantity.
What's that?
All blotting does is tell you the quantity.
So Western blotting is not real great at quantity. It's real good at yes, no, and some information about quantity. But it's not quite as good about quantity as you'd like it to be. Clear as mud? Yes?
Is it always antibodies used for Western blotting, or can you use other things that have sensitivity?
So are there antibodies always used for Western blotting, or are there other things? Antibodies are really the things that will give specificity in terms of identification of proteins. So there's other things that you can sometimes use to measure a protein's activity, but they get much more involved. So antibodies will generally be the things that's used. Yeah. Mm-hmm. How's Western blotting similar to affinity chromatography? It's not. Not at all. So affinity chromatography uses the binding of a molecule by a protein as a means of separation, and Western blotting is using an antibody that recognizes a specific protein as a way of determining if it's in the mixture or not. So they're not at all similar. Yes?
Gel filtration?
Yes.
Force that separates gravity?
In gel filtration is the force that separates gravity. That's actually a pretty good question. Okay. In a way, yes. Because with gel filtration, what you're doing is you have a column that's got these little beads with the holes and so forth in them, and you have a buffer that's passing through there, and the buffer is being pulled through by gravity. You don't pump it, for example. So since the buffer is being pulled through by gravity, and since the proteins are dissolved in the buffer, gravity ultimately is that separation mechanism. Yeah. Yes?
In the gel filtration, why is it that or do all the small portions go through the beads? I mean, why is it that they prefer to go through there and not go around, say, another one's going in at that moment?
I'm not sure I understand the question.
With the gel filtration,
Uh-huh.
that's when you're separating out, it separates out the smaller ones
Yep.
going through. Why is it that they only go through the tunnels of the bead, not both as accessible? Like, if there's room for the larger molecules to go around the beads, why aren't the smaller molecules?
The smaller ones are going around the beads, but they're also going through the beads because they can. So,
They are in both.
so the only point about the small ones is they're taking a longer path. So they can go through the tunnels. The large one can't. So because they can, they do. Right? So because they go through the tunnel, they take a longer path to get through than the large ones, which just go right straight through.
Do some of the smaller ones go straight through with the larger ones?
Some of the, well, all these are a measure of likelihood. Okay? So you never say never, you never say zero concentration. But 99.9% of them are going to not be. I mean, they're going to be back in the main peak with the things that travel through the beads. Okay? Did I answer your question?
I guess I'm just trying to figure out what gets them to go through the beads instead of straight down.
Because they can.
Because they can.
Well, I mean, so if you think about it, that let's imagine that here's a column and it's got holes in it, right? If I said to you, I'm going to put my solution on the top of the column, and it's got to travel, through this solution of buffer. Forget about the beads, just got to travel through the solution of buffer, and it's got to go down through here, right? It's going to take X amount of time for it to just go through this column, moving like I just showed you, right? If I took the same thing and I made a narrow column, there would be less volume, and the same gravity, it's going to go faster, right? So if we compare the large proteins to the small proteins are going through that larger volume because there's holes in the beads they can go through. The larger ones in the same column are going through the equivalent of a small column because there aren't all the holes in the beads they can go through, and that's why they go shooting through fast. So that's the difference between the two. There's actually a volume of buffer that they're traveling through. And the volume is different for the two. It's greater for those that can get into the holes. Okay? Yes, back in the back. Can I what? Can I talk about chaperones? Okay. So chaperones are important proteins because they help proteins to fold properly. Okay? And let's see, where did I talk about those? That was up here. I thought I had a figure, but I guess not. Okay, so we know that folding of proteins is essential for their function, and that the proper folding of proteins is essential for their proper function. So when we look at a prion is a protein that has not folded properly, and because it is not folded properly, it actually induces other copies of itself to not fold as well. Somebody asked me on email, does a prion protein only misfold copies of itself, or can it misfold other proteins as well? And the answer is it only misfolds copies of itself because it's forming a sort of a polymer, and that polymer requires everything to be the same. Okay? Misfolding, you can imagine, therefore, is very problematic. So cells have things called heat shock proteins, and heat shock proteins are induced upon the shock or the increase in temperature that a cell encounters, and the heat shock proteins are known to be chaperones. So what the chaperones do is they provide a sort of a chamber into which a protein is inserted as it's being made. Okay? And the reason for that is as a protein is being made, proteins have hydrophobic amino acids associated with them. And those hydrophobic amino acids don't like the water solution in which they're sitting in the first place. Okay? And they like to associate with other hydrophobic side chains. Well, that's fine if the only hydrophobic side chains it can interact with are those on the same polypeptide chain that is being made. However, proteins are being made simultaneously. So a protein that's being made over here that has hydrophobic side chains is right next to another protein over here that's being made with hydrophobic side chains. The hydrophobic side chains can start interacting between the two proteins, and now you have a problem. Okay? So the chaperone is there to protect and keep those guys from interacting with each other. Instead, they get secreted into this little chamber where they can only interact with themselves. So that's one of the things that the chaperone does. Okay? It allows a protein to fold on its own and not be affected by other proteins that are there. Now, chaperones may play some role in refolding or properly folding of proteins after they have gotten misfolded. That doesn't work for a prion, but it can work for some proteins after a heat shock, for example. Okay? So that's what chaperones do. Clear as mud? You guys getting hungry yet? I think beer at Bombs Away sounds really good tonight. Tuesday. Oh, question over here.
One last question. You used the word perfection yesterday to describe an enzyme that can work at the rate of diffusion.
Yes.
Why not use perfection to describe an enzyme that works at the right rate and not, the fastest possible rate?
So why you're questioning why I use the word perfection?
Yeah.
Well, I didn't come up with the term in the first place. So perfection, I think, actually is quite appropriate here because here it's perfect because if you make any changes, you make something that's less efficient. So to me, that's a very good description of what perfection actually is. So yeah, it's a semantic thing. Yeah.
I'm just thinking like the context of the cell and the enzyme. If you needed to go slower than as fast as possible.
Well, it depends on what your idea of perfect is, right? So yeah. It's a semantic argument. Okay, any other questions? Yes?
I know this is going way back to the hemoglobin thing, but how exactly, at what point does CO2 enter the equation, and how does it not become an issue with CO2 competing with oxygen? Or does that become an issue?
CO2 does not compete with oxygen and hemoglobin because they bind to different sites.
Okay, but can they both be bind simultaneously and stop?
Well, so yes, but look at what I showed happen when CO2 binds. What happens when CO2 binds? I didn't talk about T state. I just showed that it favored the release of oxygen.
There's release of oxygen.
Right.
So release of oxygen doesn't always mean T state? I guess
That's a subtle argument, but I'm just saying I didn't say T state. Right? Okay. So I'm not going to ask you to determine if it's a T or a partial T or whatever. The point is that your question had to do with can CO2 and oxygen both be bound? The answer is yes, but you see that CO2 is going to favor the release of oxygen. Right? That's the Bohr effect. Okay? All right, guys. Study hard. Stay away from those brownies.